Hello friends, and welcome back to Nanotech. Today I will discuss the impact of microgravity on human cancer cells. Gravity is something we are all aware of, an ubiquitous force that affects all living things on the planet. The significance of gravity became evident after man walked on the moon in 1969. Astronauts underwent a variety of physiological alterations while in microgravity, the state of the space. In the early 1970s, the U.S. Skylab program conducted the first experiments on the effects of microgravity on human cells. The goal of these experiments was to investigate the physiological and biomedical effects of simulating microgravity on Earth. The findings revealed that all biological living systems, whether a complex organism or a single cell, are influenced by and adapt to their surroundings. WI-38, human embryonic cells were cultured in the Skylab 3 for roughly 28 days, with the results revealing that the growth rate, chromosomal banding, mitotic index, and cell cycle were all comparable to human cells on Earth. Microgravity changed cell metabolism, as seen by increased glucose concentrations in the Skylab 3 cell-cultured media. Furthermore, both Skylab 3 and on Earth cell culture media have different extracellular matrix, cell polarization, and cell-cell interaction. Effect of microgravity on cancer cells. Viability and apoptosis. Several studies have shown that microgravity has an inhibitory effect on cancer cell viability and growth. Cancer cells are affected by microgravity by inhibiting survival signaling pathways and inducing programmed cell death. The CAV1 protein is a gravity-sensitive protein that regulates cellular proliferation, differentiation, and apoptosis, and it decreases in cancer cells after 72 hours of microgravity exposure. Growth and proliferation. Microgravity disrupts cell cycle controlling genes and proteins, preventing cancer cells from multiplying and forming spherical colonies. According to some reports, microgravity inhibited the proliferation of lymphoma cancer cells and caused cell death. However, normal cells were unaffected. Extracellular matrix and cytoskeleton. Microgravity can alter extracellular matrix and cytoskeletal proteins in cultured cells and cause spheroid formation in some cancer cells. Spheroids are three-dimensional cell architecture formations that can, to some extent, represent the tumor's physiological condition. Modifying signaling pathways, the amount of extracellular matrix, and three-dimensional growth-associated proteins could all play a role in spheroid formation. Based on bioinformatics analysis, simulated microgravity promoted the overexpression of three-dimensional growth-related proteins and avoided excessive exogenous protein accumulation. Cancer cells appear to have remodeled their extracellular matrix and cytoskeleton as an adaptive response. That is, microgravity alters cell-cell interactions, cancer cell adhesion, migration, and invasion. Some cancer cells assemble into multicellular three-dimensional constructs in microgravity. As a result, simulated microgravity may be used as an inducer in tissue engineering and spheroid formation. Cell growth, proliferation, gene expression, soluble factor production, cell signaling, extracellular matrix production, cytoskeletal organization, and cell adhesion are all affected by microgravity. It also influences the expression of cytokines, interleukins, and other immune-related compartments, as well as activating immune cells. In terms of analytical approaches derived from space-based research, it appears that microgravity could be used as an anti-tumor technology and therapeutic method in the distant future. Thank you for watching this video tutorial. If you like this tutorial, please share with your friends and colleagues. Thank you.